Assalamu alaikum guys. So we were discussing about the enzyme characteristics of enzymes in the previous lecture, and today we'll be starting the factors um, which affect the rate of enzyme action. So actually they are very sensitive to the environment, and any factor which we will be discussing now, if the factor changes, the chemistry, like it means the structure changes, the shape changes, the chemistry or shape, so both of the words have the same meaning, of the enzyme molecule that can change, and and as a result the activity will be affected. Some of the factors that can affect the rate of enzyme actions are being discussed. Number one is temperature. So if you increase the temperature of the rate of enzyme, if you increase the temperature, it speeds up the rate of enzyme action, but only to a point. See, for example, in this picture, this is the rate of enzyme which is increasing, okay, and this is the temperature which is also increasing. So when the temperature reaches till 38 or something like that, 37, when the temperature is 37 here, it means that the enzyme activity will be at its peak here, okay. So this is the point where the enzyme activity will be at its peak, and the temperature is 37, and the temperature at which the enzyme activity will be at maximum rate is known as optimum temperature. So you must know what is optimum temperature. For our body, the optimum temperature is 37 degrees centigrade. Human enzymes, 37 degrees centigrade is the optimum temperature. It means the optimum temperature is the maximum working speed. The maximum working speed of human enzyme is 37 degrees centigrade, but when this does not happen in the like in the body, like 60 degrees centigrade and things like this, but we can test it in the lab, in the laboratory. When we test different enzymes with the increase of nature, then what is the phenomena? It is the denaturation of the enzymes and the decline in the enzyme activity, which is shown here. See, when the temperature rises to a certain limit, heat adds in the activation energy. And see, first of all, the activation energy is increased. Number two, the kinetic energy is also increased. So the kinetic energy increases, the enzyme activity increases. The activation energy is decreased. Why? Because of the kinetic energy, the heat is increased, and when the heat is increased, the activation energy is actually, for example, if the activation energy is um 5, so the heat is adding till 3 points of activation energy, so only 2 points are left which we can easily compensate. So this kinetic energy is actually adding the heat which is used in the activation energy by the enzyme, so the reactions are accelerated. Please, these terms are very important: acceleration, optimum temperature, maximum rate, specific temperature. You have to use these words. Catalyzation of catalyzed reaction, all these terms are important. But when the temperature is raised above the optimum temperature, the heat energy increases the vibration of the atoms of the enzyme, and what happens? The globular structure of the enzyme is lost, which means the chemistry is lost, and the shape is also lost. The shape is also deformation of the structure, deformation of the chemistry, deformation of the um, shape, and the structure of globular structure of the, why? Because the globular structure of the enzyme is lost. This is known as the denaturation of the enzymes. When the temperature keeps on increasing, the enzyme gets denatured. Here, when the temperature is um, at 50 degrees centigrade, here the enzyme is, see, the enzyme action is less as compared to here, okay. It results in a rapid decrease in the rate of enzyme action, and it may be blocked completely. Why? Because the globular structure is changed now and the reaction is blocked. Now see, there is a point to ponder. Birds have a higher temperature than mammals. What would happen to the activity of a bird's enzyme if it is given temperature of 37 degrees centigrade? See, the birds, whenever you touch a pigeon or any other bird, you will feel that it's a little mildly hot. It's warm, more warm than us, because the normal body temperature of the bird is more. So what will happen if you give them 37 degrees centigrade of temperature? The reaction will slow down. Why? Because their optimum temperature is some other temperature, because the optimum temperature is always the temperature of the body of the organism. Our temperature is 37.6 or 37.7, so that is our optimum temperature. For birds it may be different, it may be more, it may be higher. Next is substrate concentration. So the factors which affect the enzyme activity, number one was temperature, number two is substrate concentration. If it means substrate means anything on which the enzyme is going to act, and concentration means quantity. So the quantity of any substance on which the enzyme is going to act is known as the substrate concentration. If the enzyme molecules are available in a reaction, it increase in see if the enzyme is available and we increase the substrate concentration, what happens as a result? Enzyme concentration is kept constant and the amount of substrate is increased, and a point will come where when you keep on increasing the substrate, it will not increase the rate of reaction. Why? Because see, for example, if this is your enzyme and these are all your enzymes, okay, so when you keep on adding the substrate, it will bind here, it will bind here, here, and then here. Now see, all the active sites are already bound. Now when you keep on adding the substrate, what will happen? It will not increase the reaction, it will not catalyze the reaction, it will not increase the reaction rate. Why? Because already the enzyme, the active site of the enzymes are bound and they are working, okay, so they are already occupied. Any more substrate molecules do not find free active sites. This state is known as saturation. This state is called saturation of active sites and the reaction rate does not increase. So in the previous factor we studied about the denaturation of the enzyme, and now they are explaining the saturation of the enzyme. See, this is the diagram, the graphical representation. When you keep on adding the substrate concentration, this is the rate of enzyme reaction. From here to here, what happens? All the active sites are occupied. So here all the activation sites are not occupied, not occupied, not occupied. Why? Because the rate of reaction keeps on increasing. But when it reaches this point, now all the reaction sites, all the active sites are occupied, and now the rate of reaction will not increase, it will be uniform, it will not change. Now next is the pH. So actually if there is less substrate, the reaction will be at a slower speed. More substrate, the speed of the reaction will increase. But if it keeps on increasing, then what happens? Saturation. Okay, next factor is pH. All enzymes work at see, you would have studied about the pH in your previous class. Actually pH is the level of acidity and basicity of a substance. So till 7, it actually starts from 1 and it ends at 14. So from 1 to 7, things from 1 to like 6, the if the pH of a substance is from 1 to 6, it means it is acidic. At 7 it is neutral. The example of neutral is water. And then when it starts increasing, 8, 9, 10, 11, 12, 13, 14, from 8 to 14, the substances start becoming basic. Now I think I revised the pH range, you would be remembering about it. So all the enzymes work at a specific pH, okay, each and every enzyme has a different pH, and that is known as an optimum pH. No, the maximum at, see, there is a narrow range of pH, means for example, the enzyme may work at a pH of 6.5 to like 5.5 to 6.5, so it is a very narrow range. This is an acidic range but very narrow range. So they have their narrow range of pH, and they work maximum at this narrow range. I gave you 5.5 to 6.5. Okay, so we call that range as optimum pH. A slight change in the pH causes retardation in the enzyme activity, and it may block it completely. Now see, same thing, when the pH is changed, the enzyme it causes retardation in enzyme activity, and the enzyme will not work and it will block it. Every enzyme has its specific optimum pH value. For example, pepsin works in acidic medium. For example, low pH, which I told you, like 2 3 1 2 3 4, that is low pH. So pepsin is an enzyme which is present in the stomach. It works in a pH which is low, while trypsin, again, this is an enzyme, it is present in the small intestine, and its activity is highest in the alkaline pH, which means higher pH, like after 7, 8, 9, 10, 11, 12, 13, 14. So trypsin works in higher pH, which is 12, 13, 14. Change in pH can affect the ionization of the amino acids at the active site. Now this is the reason. This is the reason which causes the retardation of enzyme. Why? Because the amino acids of the, I have explained this, that the enzymes are made up of amino acids, and the ionization of amino acids results in the change in pH. No, the ionization of amino acids results in the retardation of the enzyme activity. So the enzyme will not work when the ionization of the amino acids is seen, and that happens because of the change in pH. Again, see, this is a graph which you can understand. Optimum pH has enzyme activity at its highest here, and then it starts declining. Now we are going to discuss about the mechanism of enzyme action. When enzyme attaches with a substrate, there is a temporary ES. See, this is the enzyme E and this is the substrate S. Then what happens? An ES complex, enzyme substrate complex, is seen, and then after this complex, the enzyme is released. As I told you earlier, that the enzyme will not be used, it will be released, and the product is now produced. Okay, and an enzyme and product both are released. In order to explain the mechanism of enzyme action, who discovered it? A German chemist, Emil Fischer, in 1894 proposed the lock and key model. This model, according to this model, the enzyme and the substrate, they have specific shapes. So see, this is the enzyme. It has a specific shape, and it will be attached to this substrate, which is again very, very specific. Here it makes enzyme substrate complex, and then the product is released, and the enzyme is released as it is. It is released as it is. It is not changed. This is now, and this is the active site of enzyme. Here the substrate attaches here. ES complex is formed, and then the product is released, and the enzyme is released as it is. So now you understand what is the lock and key model of enzyme action. He um, he gave the idea of the specificity of enzyme and the substrate both. He said that they have specific shapes that fit exactly into one another. Okay, and this model, what is the significance of this model? It explains the enzyme specificity, that the enzymes are specific for a specific substrate. In 1958, an American biologist, Daniel Koshland, he suggested the modification of the lock and key model, and now he proposed induced fit model. Why induced fit? He stated that the active site is not a rigid structure. Now he said that the enzyme is not specific, it can change. So it is molded. See, the word molded is used into the required shape to perform its function. Okay, induced fit model is more accepted than the lock and key model. They can ask you the significance of lock and key model in the exam, and they can ask you the significance of the induced fit model also. So what is the difference? The induced fit model states that the active site may change. See here, the active site may alter itself. See here, it is a little bit changed, and here now the enzyme, the active site is changed. Okay, so then the substrate is same, the substrate is specific, but the active site changes and the enzyme is now having a different structure. This is known as induced fit model. It means that um, the enzyme induces and change it, the substrate induces the enzyme to change the shape.